amazing people, and also gotten to see a lot of really incredible wildlife. All of this to say, I'm basically a kid that never grew up, and I feel so lucky that I've been able to pursue my childhood obsessions through scientific research. I just wanted to add one more final word about science and people. So science is never done in a silo or in a vacuum. Everything I've ever been able to accomplish has been a result of the relationships that I've built with friends, colleagues, lab mates, and mentors through the years. So if any advice to give, it's this. It's first to maintain and foster really good relationships. Some of the best ideas have come through working with other people, and often these are the projects that end up being the most fulfilling. So the second is to seek out good mentorship. Find a mentor that will push and challenge you as much as they will support and guide you. It'll not only set you up for being successful, but it will hopefully give you the tools and wisdom to someday be yourself a mentor to someone else. So that's the story of how I got here. I hope if anything, you found the story somewhat amusing and perhaps can get some insight into one of the many pathways one can take to getting involved in research in biology and becoming a biologist. So this is moving on to the second part of my research talk. So this will be a much more classical, like here's the project that I'm doing. And now I'm going to talk to you about my current research on weaverbird nests and weaverbird evolution. So first of all, some of you might be wondering how a kid obsessed with lizards decided to spend 5 to 6 years of their life dedicated to studying bird nests. In 2016, I went back to my undergraduate roots and started working started my PhD at UC Berkeley, working with Dr. Rory Bowie. So Rory is the faculty curator of birds in the Museum of Vertebrate Zoology. He's actually the same professor that taught the ornithology section of that natural history of the vertebrates class that I talked about as an undergrad. Rory himself is originally from South Africa and has spent most of his career studying phylogeography of African birds and organismal systems there. So that's why I wanted to work with him. Even before I officially started my PhD, my mind had already been going back to those phenomenal weaver nests that I had seen in Namibia. It turns out that sociable weavers were just one of over 100 species of different weaver species that comprise the family Ploceidae. And at the time, no one had done a phylogenetic study on the group. So both Rory and I decided to have me tackle the weaver phylogeny for my dissertation. So that first fall, I requested dozens of tissue samples from different museums and immediately got to doing lab work. I thought to myself, I'm hitting the ground running. This is going to be great. Maybe I'll finish my PhD in 3 years. And then something happened in December of 2016. A paper was published by De Silva et al. Where they released the first weaver phylogeny. For the scientists in the room, you know what that means. That means you get scooped. So suddenly all the work that I had been doing immediately went down the drain, and I had to come up with a different project because I had been beaten to the punchline.
Absolutely.
So after I got scooped, I spent the next year or so trying to come up with different projects. I thought about working on everything from avian disease, perhaps another group of birds, but my mind kept going back to weavers. I really wanted to work on them. During this time, I was also studying for my qualifying exam. For those of you who don't know, the qualifying exam is this 3-hour oral examination where a committee of 3 or 4 professors just grill you on your academic expertise. And at the end of the exam, they take a vote and decide whether or not you get to stay in the PhD program. So it's a little stressful. For our program, you also need to have your dissertation proposal ready at the time. So you need to formally decide what your project is going to be about. So there was a little bit of added pressure to pick something to decide to do my PhD on. I had actually considered working on weaver nests, but based on an initial search of nest specimens in online museum catalogs, it just didn't seem like there was much out there. Even our own museum only had 1 weaver nest. But then at some point it occurred to me that our nest collection wasn't published in our online catalog. So most museum collections have been around much longer than the internet, and a lot of their catalogs were originally put together by this card system, this index card system. And so it's taken a while for a lot of museums to take those index cards and move them to an online database. And nests, because they tend to be so underutilized in museum collections, are one of the last things that get moved to an online format. And so once I had this epiphany, I ended up emailing a bunch of curators and just asking them, do you have weaver nests? And what I got were just tons and tons of Excel spreadsheets that showed that actually there were hundreds of weaver nests out there that no one had touched for a project. So suddenly I had a project and I decided, great, this is what I'm going to work on. I'm going to look at the evolution of design in weaver bird nests. So I took my qualifying exam. I passed, as evidenced by the bubbly and happy smiling faces of my committee members. And after the celebration, Steve Beisinger, who is now one of my dissertation committee members, asked me if I had ever heard of the Western Foundation. And I, because I had a background in herpetology and wasn't as in tune with the ornithological community, said, I've never heard this place. And lo and behold, there was this gigantic, one of the world's largest bird egg and nest collections in my home, Southern California. So I reached out to Linnea and Renee and asked them about their weaver bird nest holdings, and they had, over 200 weaver bird nests that I could include in my studies. That was just exciting and fantastic. So before I go into my project, I wanted to talk a little bit more broadly about bird nests. So, one might ask, like, what is the point of studying bird nests, and what is the meaning behind the work that I'm doing on them? So I kind of hinted at this already, but nests are one of the most understudied components of avian life history. This is despite the fact that they're ubiquitous and diverse. Nearly every single species of bird makes them. They play an important life history role, so they're involved in offspring rearing and in mate choice. And the other thing that's crucial about them is nest building is an innate behavior. So a bird can build a nest without having been taught by a mentor. They just know to do it without any kind of external stimulus to begin that kind of behavior, which is amazing. So all of these factors really make nests ideal for comparative studies of avian evolution. Previous evolutionary studies of nest design have resulted in two major prevailing schools of thought. One is that nest design appears to be constrained phylogenetically, even among diverse clades. To put that in layman's term, it means that even within a family of birds that are very ecologically diverse, so if they show up in many different habitats, the overall design of the nest is mostly the same. And this has been shown in, for instance, a lot of passerine songbirds. A lot of them make just simple cup nests, and whether the bird lives in a desert or on a mountaintop or in a grassland, they all tend to make cup nests. But then the alternative theory is that natural and sexual selection can actually drive variation in nest design, that maybe the problem isn't that the nests don't vary, but they vary in very specific ways. So to be able to capture that variation, you need fine scaled measurement data to be able to pick up any of that signal. So my goal of my project was to test these two alternative hypotheses, whether or not microevolutionary patterns or family level patterns of nest design match the macro evolutionary patterns of nest design that these other broad scale studies have picked up on. So this brings me to weaver birds. So the sociable weaver, which are the birds in the bottom left, represent just one member of the super diverse family of sparrow-sized songbirds. So they actually have a wide variety of different plumages and colors. They occur in many different types of habitats. They sing different songs. They also exhibit different types of complex behaviors. But by far, what makes them the most interesting are the nests that they build. Weavers, as their namesake implies, build these extremely elaborate nests by intricately weaving together nesting materials, creating a variety of nest sizes, shapes, and compositions between species. Some nests have elaborate entrance tubes. Some can range from the size of a football or softball, and others, like the sociable weaver, can get so big that they can take over entire structures. So given this diversity of this widespread and species-rich radiation, they're really this ideal system for studying and understanding the selective forces that have quite literally shaped bird nests. So for my doctoral dissertation, I've identified three primary areas of study with which to answer my main research questions. To start, what is the diversity of nesting structures in weavers? How do nests vary in size, shape, and material composition between species? The second goal of my research was actually to revisit the weaver phylogeny. So even though one had already been published by the De Silva group, their data set was a little bit limited on both their taxon sampling and in their genetic marker sampling. So we decided to revisit the relationships using whole genome sequence data. And then finally, the last goal of my project was to really get at the evolutionary history of nest design in weavers. To start, like, what is the ancestral nest form? When did weaving evolve as a nest building technique? And also, given how conspicuous the entrance tubes are, do these tubes represent a derived or an ancestral character state? And how many times have they been gained or lost through evolutionary time? To address these goals, I've collected 3 types of data that extend from specimens in museum collections, combined with additional character data from the literature and online databases, and by updating the weaver phylogeny using, as I said, advanced molecular methods. This data was then used to run phylogenetic analysis of character evolution using some methods called ancestral state reconstruction and stochastic character mapping, which, again, for the non-scientists in the room, I promise I'll explain once we get to those parts of the talk. The first museum I visited was actually the Western Foundation, given its location in Southern California. This first trip was actually more of a reconnaissance mission because there was no guidebook for how to measure bird nests. I ended up having to come up with my own protocol for pulling measurement data from every single species. This is actually a harder task than you would think. Nests are fragile, they're irregularly shaped, some nests are incomplete or damaged, and the way that they've been collected varies from specimen to specimen. So this first trip was extremely valuable because I ended up coming up with a protocol that involved using digital calipers, tape measures, and also using a camera to take macro photographs of the nesting materials in order to get at things like material size and density. I also got to see in person some of the amazing diversity in weaver bird nests and how beautifully structured some of them were. They really are like these little works of basketry art. So it was really great to just have that experience of putting specimens in front of me and just starting to get a feel for what the diversity was. The next place I visited was the Museum für Naturkunde in Berlin, where I worked with Pascal Eckhoff. Working in Berlin had its other perks beyond just having a great collection at hand. I was able to ride my bike to work every day. I got to see the Berlin specimen of Archaeopteryx. For those of you who don't know, Archaeopteryx is known as the first fossil bird. It has obvious transition. It's an obvious transitional specimen between dinosaurs and modern birds because it has a beak, but it also has teeth. It's got this long bony tail and wings. And because of how complete this specimen is, it's often been dubbed the Mona Lisa of fossils. And so they have it right on display in the main gallery. So it was really cool to get to see it. So the second museum I visited was the Natural History Museum in London, where I work with Douglas Russell. The bird specimens, including all of their nests and eggs, are actually located in another facility in a town called Tring. This is because during World War II, when the city of London was being bombed by the German Blitzkrieg, the curators wanted to protect these specimens from being destroyed, so they moved them all to this other facility. Again, their collection is incredible. They have over 200 nest specimens, and I ended up having to spend 2 months there collecting data, which I know 2 months in London sounds like really rough field work. I know. It was great. So after I visited those 3 museums, I ended up with a final sample set of data from 580 nests from 84 different species, capturing about 73% of the family diversity. This is just a brief outline of all the measurements that I took, in case you're wondering, like, what did I actually collect from the nest specimens? So shape and size measurements were taken directly from nest specimens. So this included things like nest shape, length of the tube, nest length, width, and height, the size of the entrance to the actual nest itself, And then you can also get some idea for how the nest is constructed by looking at the way that materials are put together in the nest itself. So also just to point out, most species of weavers weave the nest, but there are some species that don't weave. They actually build the nest by thatching pieces of the materials together. So this is another thing that I was scoring as a categorical variable. I was also able to take measurement data on the material size and density from macro photographs. So you take a bunch of images of the nests, you put them into a piece of software, and then you have an army of diligent undergrads go and measure the size of grasses for a few months. So I'm super grateful to all my undergrads that did a lot of this work. But from their efforts, we were able to get 36 size, or sorry, we were able to get measurement data or material measurement data from about 475 of the 580 nests that I collected other data from, which is pretty phenomenal. I have a lot of data, obviously. So for today, I'm going to be focusing on the 3 discrete characters that I looked at within a phylogenetic sense. I'm just starting to analyze my continuous trait data, so that's why I'm going to focus on these for today. But I will get into some of the continuous variables when I talk later about perhaps the role of life history in the evolution of nest design. Before I get into my analyses, I did want to provide everyone here with a basic introduction to phylogenetics, since I'm going to be talking about phylogenies and showing you a lot of graphs that if you don't know what a phylogeny is, it's going to mean nothing to you. So as I said, I study phylogenetics, and the way that we present our data is through this figure called a phylogeny. A phylogeny, or as others call it, a phylogenetic tree, is just like a genealogical tree can show you relationships between family members. A phylogenetic tree is a visual representation of the evolutionary relationships between different species. When you're looking at a phylogeny, the tips of a tree represent current species, and the nodes that connect them represent hypothetical ancestors. The branches themselves represent time, so the longer the branch length, that means the longer the time has passed between ancestral splits in the current species. So if we were looking at this tree on your left, the way to interpret this phylogeny is A and B are more closely related to one another than either are to C and D, and all 4 of them are more closely related to any of them are to E. In the past, before modern genetics, typically morphology was used to construct these relationships, usually by trying to find similarities between characters such as scale counts or bones. Today, DNA sequences are primarily used to reconstruct the relationships, and the reason we can do this is because DNA is inherited, and over time, DNA tends to accumulate mutations in their sequences. We as scientists compare these mutations using phylogenetic tools and statistical analyses to reconstruct the relationships. Similar to using morphology, organisms that are more closely related to each other have greater similarities and fewer differences in their DNA sequences. The reason this molecular approach is especially powerful is because often some relationships can be nearly intractable to resolve with morphology alone. A great example is like the relationship between birds, turtles, and crocodiles. For the first time, there was debate on whether turtles or crocs were the sister taxa of birds. And by using DNA sequences, scientists were able to establish that in fact, crocodiles and alligators are the sister group to birds. I also wanted to give you a breakdown of how one does a phylogenetic study. Just to prove to my family that I have indeed been doing a lot of work throughout my master's and PhD. So there's a lot of work that goes from a sample to an actual phylogeny. So when you're doing a phylogenetic study, you usually start by picking the organism that you're interested in working on. So it can be a vertebrate, it can be a plant, you can even study the phylogenetics of viruses and bacteria. You then have to choose the appropriate markers for your study. So this can be individual genes, or because it's getting increasingly cheaper to do so, you can now just sequence entire genomes, which is all the DNA contained within an organism. You then do your lab work. So you have to get in the lab, you have to extract your DNA, do all the DNA sequencing. You get your DNA and you use software to create a DNA alignment. Once you've made your alignment, you can then run all of your phylogenetic analyses. So these are your statistical tests. Yeah, so I'm good. That's definitely showing. Okay, so you do your model testing, you do your phylogenetic analyses. These are usually broken down into two types of analyses. So you can either do maximum likelihood or Bayesian. It's just a different statistical approach for inferring the relationships with your data. And then finally, when you're done and you have a phylogeny, you actually have to come up with some way of measuring how robust you think the relationships are. So you have a hypothesis of what you think the relationships are between each species, and then you have to run secondary analyses to assess the strength of the relationships. What's the probability that these relationships are true and accurate? These probability scores are usually represented as bootstrap values or posterior probabilities. I'll have some of these values on my slide, so that's why if I say something about it having a high support value, that's what I'm referring to. So moving on to revisiting the Weaver phylogeny. So as I said, the De Silva group had already done the Weaver phylogeny. This is a representation of their tree. And so even though they had made this first assessment, I wanted to revisit it to see if we can improve on some of their work. And the reason being that when you're doing analyses of character trait evolution, the way that I wanted to do with nests, these are entirely dependent upon robust and correct phylogenetic inference. The previous work, as I said, De Silva had done their previous study, but there were some relationships that still were unresolved, especially within the genus of Euplectes, bishops, and widowbirds, and within the true weaving clades. That's one other gigantic genus within the Weaver family. So to do this, we used a whole genome sequencing approach. We sequenced 63 species of Weaver birds on an Illumina NovaSeq 6000. So it's just a fancy type of DNA sequencing machine that does this. We were able to get half of the family diversity. So all of the little tips that have been starred are all the species that we're able to get whole genome data for. We ended up using 5 nuclear and 5 mitochondrial markers, which allows us to combine our data with available GenBank sequences. And then I did all of the normal phylogenetic analyses that I walked you through before. I ran maximum likelihood and Bayesian reconstruction. And yeah, so those were all the methods that I used to make our phylogeny. So when I was done, our final phylogeny had 199 samples from 109 of the 117 known Weaver species. So I'm showing you here the top half of our reconstructed phylogeny. Overall, we were able to retrieve higher nodal support values and found that the deeper splits among early diverging lineages were largely congruent with different studies. But we did find some topology differences within the Euplectes bishops and the widowbirds, which I've highlighted here. So these are all, everything that I've starred were novel relationships that we retrieved from our own analyses. And we also found differences among the African Ploceidae. So this is the large group of what we call the true Weaver clade. So these are all the other relationships that differed. So again, it was good that we ended up redoing this because we did find some different results. And so taken together, we find that increased taxon and character sampling helped us resolve some of these relationships that De Silva was unable to resolve themselves. So moving on to ancestral state reconstruction analyses, I also wanted to provide a brief overview of what an ancestral state reconstruction analysis is. So the purpose of an analysis is to understand patterns and timing of character evolution across the tree of life. The general approach is you take measured characters from extant or living species. These are the character states at the tips of the trees. And then you use phylogenetic methods to infer the ancestral character states, that is the character states, the nodes between the species. This allows you to ask questions like, what is the ancestral origin of a character? How many times has a character been gained or lost through time? And which characters are derived versus ancestral? So in this figure here, we see 4 flower color types. And so there are 4 color types based on the distribution of colors at the tips. The analysis would probably point to red being the ancestral color type, with purple and yellow representing more derived character states, with red having been lost twice, maybe 3 times. And so the probability of an ancestral node being a character is usually depicted in these pie charts or these pie graphs. So that's typically how these analyses are depicted, to give you some sense of confidence in the character states that you're estimating. So with my own analysis, the first character that I analyzed was nest shape. We had 5 nest shape categories that were adapted from Crook et al. So an earlier study was done on just the morphology of one group of weaver bird nests, where they tried to define nest shape using different types of shape categories. Our own categories included compound nests. So these are nests that are constructed of multiple different compartments, kidney-shaped nests, retort tube nests, which basically look like kidney nests with the tubes added, simple globular nests, and then a specialized nest form called retort funnel. When I ran my ancestral state reconstruction analysis, we found these were our major findings. So the ancestral Ploceidae nest form is likely globular. We see repeated evolution of various nest forms across the phylogeny. There's at least 2 independent origins of retort tube nests in Asian and African Ploceius. And we also find, interestingly, the retort tube nests are actually evolutionary precursors to tubeless nests. So that means you have a secondary loss of tubes in the African Ploceius. The next character that I looked at was the evolution of nest building, the nest building technique. So in this analysis, I categorize nest building techniques into 3 categories. So either nests were thatched. So this is, again, just the simple insertion and separation of compacted nesting materials. Nests could be constructed through a minimal weave. So in this case, a bird may take a long strip of grass, make all these really intricate knots and trellises, but the knots and trellises themselves are confined to the growing edge of the roof or wall of the attachment. There is minimal cross-trellising across the actual body of the nest itself, and this can usually result in either a tightly or loosely woven nest. The last category was a complex weave. So in this case, you have extensive knots and loops, stitches throughout the nest body. In all cases, this results in a tightly woven nest. So when I did my ancestral state reconstruction analysis, I found that all early diverging lineages construct their nests by thatching. So thatching is an ancestral behavior to weaving. We also find a single origin of weaving between 7.5 to 10 million years ago. And then finally, we find 2 transitions from a more complex weave to a minimal weave among Foudy and Euplectes. So complex weaving is actually the precursor to a more minimal weave, which is also interesting. The last character I looked at was the evolution of the entrance tube. So in this case, the character was a lot easier to score. Either a tube is present or it's absent. When I ran the analysis this time, what we found was that the ancestral nest form was tubeless, which is not really surprising, and tubes themselves are a derived character state. The other thing that we see is we find multiple gains and losses of tubes throughout the family's history. And we also find that the ancestral African Ploceius melimbus nest was likely tubed, followed by 3 losses in its evolutionary history. What was also interesting was that tubes are not actually restricted to just the retort nest. They also appear to be present on the compound nest as well, which you can see by the black circles in the top half of the phylogeny. So this is just a summary of the research that I've done so far. So we find that the use of whole genome sequencing data greatly improved character and taxon sampling, leading to better support across the phylogeny of the weaver phylogeny. Currently, further analyses using our whole genome sequence data are ongoing. And so I'm hoping to work more with this genetic data before my dissertation ends. The other thing that we find is that the ancestral weaver nest was likely a simple globular nest that lacked a tube, and that thatching as a nest building technique is ancestral to weaving. We also find repeated evolution of various nest architectures across the family, which means that, again, this is taking us back to that initial set of hypotheses that I presented you with. So this means that nests themselves are a lot more evolutionarily labile than one would necessarily think. So they're not constrained by evolutionary history. And in fact, it seems that nests themselves can be somewhat adaptive, and natural selection can actually drive variation in nest design between different species. What's more, previous family level studies tend to show that nests become increasingly complex through evolutionary time. Early studies on swallow and ovenbird nests by Winkler and Sheldon and Zykowski and Prum, respectively. In their studies, they found that closed nests darned with roofs are derived from open character states with no reversion back to ancestral morphologies. However, in this study, we find that some complex elements like entrance tubes and the weaving technique can actually revert back to more simple states. So given this reversion, we need to investigate more life history effects, which is what I plan to focus my studies on moving forward. I've already started to get at this question of the role of life history in nest design, but looking at variation design between some of the subspecies that we sampled. So weaver subspecies can differ in body size, in geographic locality, in their habitat, and in clutch size. So this gives us an opportunity to potentially identify some of the major factors that may be contributing to differences in nest design across the weaver family more broadly. The first subspecies that I looked at were the two subspecies of Ploceidae nigerimus. So this is Veliot's weaver. There are two subspecies. The first is Castanio fuscus, which occurs in Nigeria, Sierra Leone, Ghana, Togo, and Liberia. And then there's nigerimus, which occurs in Uganda. When I ran a PCA on my data set, so PCA analysis allows you to take all of your data and figure out where is most of the variation coming from, what's actually providing some signal of variation in your data set. So when I ran this type of analysis, we found that nest height and width made up the first principal component, while nest length made up the second principal component. This is interesting because this might suggest that height and width are separate evolutionary modules from length, and they're not evolving together. I also wanted to point out that material size and density was an important loading factor on principal component 3. So it does seem like there is detectable variation in material size and density even between the subspecies. I then ran an ANOVA, which is an analysis that allows you to say, so you find that there's variation, but then you want to ask, is this variation significant? Does it matter that there's variation? So I ran an ANOVA, which is that type of analysis, and we found that out of the 3 principal components, only nest length appeared to show significant variation between the subspecies. And when I plot this on a graph, so you can see that the nigerimus is in that bright pink circle. So they're occupying slightly a different part of morphology space than the other subspecies. And this is mostly because the nigerimus nests are longer than Castanio fuscus. So you might be asking yourself, why are the nests longer in a different subspecies? I looked into this sort of cursorily. I looked up some of the data that was available between them, some life history data, and I did find that nigerimus can actually weigh more than Castanio fuscus, so up to 44 grams for adults versus 39 grams, and that their egg dimensions are actually average slightly larger than the other subspecies, which is interesting. I also looked at one other set of subspecies. So I looked at the subspecies of the village weaver. So that's Ploceidae cucullatus. The 4 subspecies that I looked at were Ploceidae cucullatus cucullatus, Ploceidae cucullatus frobeni, Ploceidae cucullatus nigriceps, and Ploceidae cucullatus bondorfi. There was no cute little bird figurine available for bondorfi, so there's Bondorf. So again, I ran my PCA to find out where was a lot of the variation coming in this data set. And again, I found that nest height and width actually appeared to be loading again together on principal component 2. And that nest length was again coming out on a separate principal component. So again, this kind of shows some way, and I actually looked at this in other subspecies and I got the same pattern consistently. So it does seem that height and width is potentially loading separately than nest length, which is again to me really interesting. And we've also found in this analysis that material size appears to be accounting for a lot of variation as well. So I'm glad that my poor undergrads didn't end up collecting all this data for no reason. It ends up being actually somewhat significant. When I ran, I ran an ANOVA, which tells, this type of analysis tells you that the variation is significant, but an ANOVA analysis won't tell you what differences between the subspecies are significant. It just tells you that there's something significant present in your data set. So you have to do another analysis where you do pairwise comparisons between each subspecies to figure out where is the significant variation coming from. And so when I ran this analysis, I found things like the cucullatus cucullatus nests are wider and taller than the other subspecies. Bondorfia appears to have nests that are comprised of smaller nesting materials compared to the others. And so I was thinking about all this and trying to figure out why this might be the case. And unfortunately, as is the case with a lot of African species, there sometimes isn't great life history data at that level, at that type of fine-scaled micro level. So there wasn't really any information that I could immediately glean on differences that might exist between differences in body or clutch size. So but what I do know about the different subspecies is that they live in totally different parts of the African continent. So it might be that these differences may be related to variation habitats across the ranges of each of these subspecies. So the point of all of this is that, I ran these analyses. There does appear to be nest variation even at the subspecies level, which provides more weight to my initial insertion that there is quite a bit of variation in nest design at these micro evolutionary levels, these smaller timescales. But it also highlights the need for more nest specimens. We need greater series of nest specimens and collections in order to really capture what's likely to be quite a bit of significant variation in nest design. Also, it seems that body and egg size might be linked to differences in nest design across weavers, so that would make sense since the two things that go in a nest are bird bodies and bird eggs. So that's what I want to look into a little bit further. And then finally, I think that there is a real important need to look at the role of ecology and how habitat might play a difference. Like there might be differences in nest design pressures based on how high off the ground the nest is, where it's placed, what kind of predators are mainly putting predatory pressures on these nests. So these are the types of things that I'm working on moving forward. So with that, that's all my research. I really want to thank all the people that have been involved in this project so far. So my thesis committee has been extremely supportive, all the staff at the Museum of Vertebrate Zoology, Bobby, who's present in the talk today. A lot of the analyses that I ran, the ancestral state reconstruction analyses, he helped with immensely and providing R code and scripts. I have an army, as I said, of undergraduate assistants that have been providing me with a lot of effort, or a lot of their own efforts and support in collecting data. I really want to thank all the nest collection curators that have been helping me with this project. So Linnea and Renee, and of course, Douglas and Pascal. All of the museums that have graciously been providing me tissue samples, even during the pandemic. I've been able to receive data and tissue samples from different museums, even during this time. Of course, I have to thank all of my funding agencies. There's been a lot of funding agencies that have been providing me with financial support. And also, I wanted to take this opportunity to thank all the people, all of my family members that have been so supportive through the years as I've been on this interesting and somewhat unique career path. So, my mom, my grandmother, my dad, especially, who you can see definitely has had an impact on my love for the scaly animals. And I also really would like to thank my partner, Daniele, who helped me prepare for this talk and listened to me rehearse it a bunch of times. So thank you so much to Daniele for being so supportive. So with that, I'll leave the room up to questions. And if you want to hear more about my research, I am on Twitter, and I also have a website that you can go to that's got a list of current news and things that I'm up to.
Well done. Everybody can unmute themselves now if you would like. We can hear you. We can hear you clap. Okay, Jackie, do you mind? Let's take your screen down. Let's stop sharing just for a moment, although there might be questions that come back up where we need to go back to maybe some of your slides. So again, thank you very much, Jackie. That was awesome. Yeah, well done. And thank you for, providing information about your background too. It's wonderful to hear. So I know that I have a few questions myself, but there are a couple that have been put into the chat too. But if you want to actually ask Jackie a question directly too, I'll answer the question in the chat first, or we'll let her do that. And then, just put your hand up or something, and we'll unmute you, or you can unmute yourself, and we'll let you ask her directly. So let me read you the one though that first, Jackie, that's in the chat. So Howard Higley says, in addition to the genetic variation and nest design features between weaver species, is there any influence of colonial versus solitary lifestyle on the phylogeny of the family?
That's a great question that actually Bobby might be able to answer, because as a part of our collaboration, we decided to focus on different parts. Obviously weavers are really complicated. They've got a lot of different things going on, including their life history. And I forget who asked the question, but one thing that makes them really interesting is the degree to which they vary in their sociality. So some species are solitary, some species live in these big colonies. So I believe Bobby actually presented on this research at AOS. I don't want to throw you, I don't want to be like, here, you answer that question, but I know that this is actually something that Bobby's been a lot more focused on than I have personally.
Bobby, you want to?
Sorry, I was just unmuting. So Jackie is correct that there's a lot of diversity in both the mating systems of the weaver birds, the degree of sociality. Some of them live in these large, complex colonies. Others live in more solitary type of habitats. Some of the patterns that we've noticed with the weaver birds is that the species that have monogamous mating systems tend to have, at least in our preliminary data, globular shaped nests, whereas the species that live in polygynous mating systems, polygynous being where one male mates with many females, they tend to have some more of the more complex nests that Jackie was talking about, or either the kidney-shaped nest or the retort-shaped funnel nests.
Oh, that's interesting. Jackie, do you want to add anything else to that?
Yeah, it's funny, I was presented with this question when I gave my talk at the MVZ symposium, but it was asking whether or not there might be a trade-off between nest complexity, plumage complexity, and song complexity. And I was like, oh, that would be such a great project. So there's this prevailing theory in evolutionary biology that you get this trade-off. Either you're very sexy, but you kind of have a crappy bird song, or you're maybe not as sexy and you have a really complicated bird song. And it would be really interesting to integrate nest design into that model. To what extent, like if you have a really complicated nest, does this undermine some of the other features that are usually involved and put under pressure by sexual selection? So maybe a postdoc project, I don't know. It's a lot of data to collect data on bird songs, but there's one thing about ornithologists is that they're very organized. There's so much data available on online databases. There's actually a repository of birdsong, and I know that there's a lot of weaver songs that are available on there, so that would be a really cool avenue to look into.
There's always egg color.
And egg color.
Not as,
oh my gosh, there's so much
not as big a deal with these birds necessarily, but yeah, it's still another factor. Okay, so another question from the chat, the written chat. So, when you were talking about how the nest tube characteristic came and went, the question is, do you have a hypothesis, or are there hypotheses as to why the tubes would be lost and gained? I mean, maybe you can just address that generally.
Yeah, so you can imagine if you're building another entire structure on your nest, you're adding an additional feature. There's a cost to that, right? You have to get more nesting materials. You have to put in the metabolic energy to creating a tube itself. So there has to be, if it wasn't evolutionarily costly, then more birds would do it. That would be the hypothesis, right? And so there's different reasons. So there's not enough of a selective pressure to have them. I don't know what are the main types of predators that are associated with the presence of the tube, but I suspect it's probably related to the prevalence of avian predators. They've seen hawks try to get at these tubes, and the tubes are actually pretty strong. Like, it's hard to pull them apart.
Yeah,
And so they've seen hawks try to pull at them to no avail. The other two things are snakes. So there are actually a lot of snakes in Africa. The way that these nests are typically situated is that they usually get put at the very tips of branches. And so what will happen is a snake will come, try to navigate the branch, and then move down to try to get into the nest. But if there's a really long tube, it's a lot harder to do that. So they've actually seen snakes try to get in and then fall because they can't get up the tube. The last thing that, the other main factor are brood parasites. So one of the most common brood parasites in the African savanna are the Diedrich's cuckoo. And cuckoos heavily parasitize weaver nests, as this has been shown. And so the cuckoos are much larger than weavers. And so the idea is that the tube is large enough to allow a weaver to gain access to the egg chamber, but it's a lot harder for a cuckoo to get up something like that, especially if it's really long. So those are to me, at least those are the three main types of pressures that I can imagine would either move a weaver towards wanting to build a tube or not. So if you don't have those types of pressures, maybe there's no need to go through the extra effort of adding a tube to your entrance.
Great, thank you. Okay, so Diana.
Yes.
Has a question.
Yes. So when you were talking about the differences in material size and density in the different weaver nests, I have two thoughts on that. One is that largely based on what the weavers prefer, or is it due to local availability? And what effect is the availability of different materials on the different nest structures?
Yeah, that is a great question. Yeah. So there's a few things there. I imagine that there's some degree, so what materials are available and when has a huge impact. One of the things that I'm actually looking at with my data set is I started taking measurements on bill morphology. So I'm measuring the size and slenderness of a bill as a way of trying to get at does having a slender bill allow you to manipulate finer or larger nesting materials? And so that would, so if I find anything with that, that might suggest that there's not a restriction, but depending on your size as a bird, that might limit what types of materials you can actually manipulate. It's one hypothesis, so it's something that I'm able to look at with specimens. But of course, there's other things like what things are available, whether there is a type of preference. I know that in some weavers, and I haven't looked at the behavioral data as much, there's a preference for having fresh materials versus not fresh. So if you're going to take a long strip of grass, obviously if the strip of grass is dry, it's going to break. So in order to actually make a tightly woven nest, you need to have access to long blades of grasses that can then be manipulated and woven through a nesting structure. So yeah, I don't know if that answers your question entirely, but these are the things that I'm certainly thinking about.
Thank you.
Which of course could be impacted by climate change.
Yes.
Right.
And
so, availability of grasses, availability of habitat in general, but this is why it's so important to get to know these species and to conserve them, conserve that biodiversity and race against time as much as we can to make sure they're conserved.
Yeah, you actually bring up a good point. So this is actually kind of a sad thing that I found when I was in London. So in addition to climate change, urban development is a huge problem. So especially in Africa and especially in places like Madagascar, where the habitat is being completely decimated for like palm plantations and monocultures, a lot of the materials that some species rely on are being impacted. A good example of this are the fouties. So fouties only exist on islands off of mainland Africa, especially Madagascar. Most of them are endangered, and I think maybe one has actually gone extinct. This is partially from development, but also from the introduction of rats. And one of the things that I found there when I was looking at the Foudy nest is sometime in the 1970s from their collections, you actually start finding strips of plastic that the birds are having to use to be able to make their nests. So it was kind of a sad, like, oh no, they're running out of their normal mosses and softer materials that they would normally use. So instead they're using plastic to build their nests.
We actually found that in Guatemala over, when we worked there for a decade. There was relatively few pieces of plastic in the nests when we first started. This is Renee's project, and I was helping in the early 2000s. And by the time we were, in the middle of our project and toward the end, 70% of the nest would be made of plastic. So, there's these changes are seen dramatically in museum specimens and of course in the wild. Kathy, Jonakuchi, you have a question.
Yeah, actually, I have a couple of questions, and thanks so much, Jackie. This was a really fascinating talk. So I'm not a researcher. I'm a birder. I was a zoology undergrad at Davis, and I'm a veterinarian now. So if I sound a little naive, so how do you collect your genetic material? Is it blood? Is it feather, pulp, saliva?
So it varies from collection to collection. A lot of I didn't collect any of these samples. They were loaned to me from different museums. Most of not most, but a good chunk of the samples were just blood. So these are oftentimes, if you're not going to take a voucher specimen, you can get a bird and just get a blood sample, which you can then get DNA out of. And then I did receive some toe pads. So toe pads get taken from actual skin collections, and then you have to pull data out of that. And then finally, you often get tissue from actual organs. So when a specimen is prepared, we try to do our best to make sure that the animal didn't die in vain, that every bit of the animal is used for some kind of molecular analysis. So you can get lung tissue, you can get brain tissue. For DNA sequencing, you usually use liver or muscle. So that's typically what I'm working with.
That's really interesting to use all the different bits,
different
bits.
Yeah, different organs will express DNA differently. And so the ability to take those types of samples and actually get specific what genes are being expressed in a brain versus being expressed in the lung, you can do those types of analyses now. I'm not doing those analyses yet, but that's the type of really powerful data that you can get out of collecting that type of material.
Great. So my second part is I read about Trang in the book The Feather Thief.
Right.
And I don't know if you so I'm wondering if their security is now. Do they have bars on the window so people can't break in and specimens?
Oh my gosh. So for those of you who don't know, so The Feather Thief is actually this amazing book about the break-in at the Tring Museum that happened right before I went. So we often forget that these collections, we think of them as being really, really important for the data that they have, but there are a lot of really horrible people out the world that think like, oh, there's a museum that's got all these like really rare birds and feathers that I could potentially steal from. And so this guy like pretended to be a researcher, did like a reconnaissance mission and checked out the Tring Museum, and then later broke into the museum and stole a bunch of Darwin specimens actually and other specimens, and just what they did was horrible. But you're right, when I got there, their security protocols were super beefed up. I had to leave all my bags at the front. Like I couldn't have like pockets on me. Like every day they were like, okay, do you have any birds on you? And I was like, no, I swear I didn't take anything. So
pat down, you got the pat down.
Yeah, exactly. Yeah, but I wasn't allowed to have any bags with me at all when I was in the collection. They were very strict about when I could access things, what things I could have at. So yeah, they're definitely been impacted by that experience. And it's unfortunately kind of a warning message to all museums really, because I think there's, the idea of someone doing something like that just goes over our heads, but it's a good warning that, we really need to make sure that these things are protected from bad people.
Yeah, it happens.
Right.
Bobby,
that's
oh, sorry. I didn't mean to interrupt you, Kathy. Did you finish your comment?
No, I'm done. Thank you.
Okay. Yeah, thank you. Bobby, you had a question too.
Yes, and I want to credit Noah, who's one of my collaborators that's here. He's asked me this question before. But Jackie, David Lottie, who's also one of the audience members, has studied weaver bird nests in places where weavers have been introduced, like the Dominican Republic, for example. And he's found that there's been relaxed selection because of lack of brood parasitism. There's changes in their egg coloration. So my question is, you have really interesting results with the different subspecies of weavers and showing that there's some different patterns in the size and shapes of the nest. Have you considered possibly looking at introduced populations of weaver birds to see if nest form or size or shape has changed over time on these different islands?
So I have a few of, it's one of the Cucullata species. It's on Sao Tome. So the problem that I have with a lot of the species from the museum collections is I don't have really big series. I picked the subspecies that I did because I had a lot of individuals. Unfortunately, I think for all of the island species I have, there are only 1 or 2 nests. Like I only have, I'm just thinking off my head, the Sao Tome introduced, I think it's Cucullatus, I have 1 sample for. And even with the Sao Tome giant weaver, I think I've got like 2 samples. But I think it would be really interesting to look at that because you're right, like there's a release of like predatory / I guess brood parasite pressure to see how that impacts the design features of the nest. I know, aren't there weavers in Southern California as well?
I just had 2 at my house.
Okay. So maybe we need to get Linnea to start collecting some weaver nests for us so that we can see if there's
They were a male and a female too. So
okay. But yeah, that would be a really great avenue to look at. And if not in weavers, then maybe in another group too. Because I might continue working on nests post, we'll see. But yeah, I for me right now though, it's just an issue of power and not having a series.
You have wider birds in California too, also.
Yeah,
that's amazing. I mean, I wonder who they parasitized, but that's interesting.
yeah, well, let's hope they don't parasitize. Well, we have Munias.
Okay.
Right, so maybe they'll parasitize the Munias and keep the Munias in check, but who will keep the others in check? I don't know. So yeah, it's always dangerous when new species are introduced. David, you have a question.
Sure. And Jackie, you know, Jackie's lab and our lab are collaborating pretty closely, so I'd be pretty disingenuous not to mention that. I'd also actually be pretty disingenuous not to mention that when I just finished my PhD with Bob Payne at the University of Michigan, he said, okay, for your postdoc, till you get a real postdoc, why don't you do weaver nest evolution? And I was like, well, I want to go on to something else. But that was like way back in 2003. So that's really, you know, the dark ages now. So I'm really glad, like personally, that Bobby and you have gotten into this, and because it's sort of like, you know, I always wondered what it would have been like if I had gone down that route instead of studying swamp sparrow song after I did my weaver work. But so I'm not going to ask you a question from my perspective. I'm going to pretend that I already looked through the people, and Gary Schnell is not here. He's professor emeritus at University of Oklahoma, but he's the last person to do what you did, I believe, not including the Callias books, which are more sort of intuitive and natural history like. So in 1973, he, you know, created a phylogeny, but not like you did, that's like a real phylogeny based on genetic data or even morphological data. He did it just on the basis of the nests. Like, imagine if the nests were the only thing about this bird that we knew about. How did the nests evolve? So it's, I guess, you know, we would call it a phonetic tree. And I can't help wondering if you placed your tree based on actual DNA next to his tree based only on nest structure, based on Crook's data. So this is the stuff that you've looked at, what it would look like, and whether it wouldn't, you know, look the same at all, or whether it would, you know, whether his phonetic tree would look like your phylogenetic tree. Have you thought about that, or have you done that, at least in your head?
You know, I hadn't. I think that's a great idea. I imagine that some of the broader patterns would probably be similar, like globular versus, because a lot of the weaver, the like true weaving all kind of had that like kidney shaped nest. The only thing that might differ is how he treated the tubes. Like, did he group all the tube nests together versus not? Because of course, our finding that was really surprising was that you see tubes popping up and disappearing at different parts of the tree. And so I think that's where you get probably the most variation. But the broad patterns of nest shape, like the Euplectes tend to make a similar nest. I can look at a nest and say, that's a Euplectes nest. I can look at a nest and say, you know, that's definitely a Foudy nest. So I imagine that if the groupings were based on those shapes, he might actually have picked up a lot of the same genetic signals, because like I was trying to get at, the fine scale variation that you see is in like nest size and the density composition. Like that stuff is where you're getting all that fine scale variation. But the nest form actually seems to be somewhat constrained phylogenetically with some variation. The only one that's interesting, the only one that really breaks that rule is the Asian Ploceidae versus African Ploceidae. So for the longest time, we thought that that's why they're all called Ploceidae, right? Because we thought they were all in the same genus. But it turns out that they're not closely related to each other at all. They're distantly related, but their nests look really similar. So that's really interesting.
Yeah. Oh, that's interesting. And how about the, he concluded that woodland, you could tell a woodland species from a non-woodland species. Do you think that that's true or no? You mentioned habitat, but I can't remember whether you actually had done.
So this is totally anecdotal, and this was just something that I was thinking about when I was doing the tightly woven versus loosely woven. So remember when I was going through my evolution of nest building technique, and you have originally you get a appearance of a compact weave with a reversion to a more loose weave. So there's less, you get like messier woven nests. One of the things that I was thinking about is that related to habitat? Because if you're in a habitat where you've got more shelter, so more canopy and things protecting the nest, there's not as much of a need to create something that's really tightly compact, right? Like you've got enough protection from wind and other things that maybe you don't need to make something that's as tightly woven. And you're also sheltered from the sun, you're sheltered from the elements, versus things that live in like open grassland. This, when I think of the other weavers that you can find in Savannah, they've got their nests hanging out in these like bare acacia trees, and they're completely, subject to the elements, right? They're being tossed around by wind. There's sun beating down on them. There's rain coming on them. And so those nests, not only do they have to be compact to protect them from getting destroyed by those elements, but they're also just, they're being tied to these really tiny tips of branches. So there's a need to stay adhered to the actual attachment point itself. So that's what I was initially thinking. And that's one thing that I want to look at, whether like canopy cover is related to how tightly woven your nest is. So that's probably something that he picked up on that, again, I was just sort of thinking about when I was doing my analyses.
Yeah, great hypothesis. Thank you.
Thank you, David. Okay, I think let's do one more question. And then I have several for Jackie I will email you later. Bruce, you want to go ahead and ask a question? If you still have it.
Thanks, Jackie. It was a great talk. I'm an ecologist, not an ornithologist. So first question is, I believe I've heard that the social weaver nests are used for many years consecutively. But how about the solitary nest? So thinking from an ecological perspective and potential drivers for different nest types and structures, there's the energetic cost, and how many of these nest types are reused year after year, and how might that influence the selective drivers? And then a related question based on work with swallows and bank swallows and others, there's one of the disadvantages of longer term nest reuse is buildup of nest parasites, swallow bugs and other parasites that can get a good load going in the nest. So if the nests are reused over a number of years, might that be a factor for weaver birds?
Yeah. So you're right. The sociable weavers, they just keep adding materials on from generation to generation to generation. I don't know what the oldest sociable weaver bird nest is, but it's probably, I'm going to just take a stab in the dark, and maybe David or Bobby can correct me, but upwards of between like 50 and 75 years. Is that okay? Shaking. Okay, good. So yeah, they can be pretty old. And a lot of the like Christmas tree ornament nests, as I like to think of them, those pendulum nests, especially in the large colonies. So in a lot of the species, the males are polygynous, and they're building not just one of these nests, but building many of them during a breeding season. So I think in the village weaver, I think the highest number of nests that was recorded for a male was like 17 nests in one breeding season. And part of that is the male builds the nest, a female checks it out, and if she doesn't like it, she'll like destroy the nest and have to start over and build another nest that's more to her liking. So they're constantly having to rebuild more and more of these nests during the breeding season. There's probably variation in this too. Bobby, you might have to like save me for a second, but I can't remember whether or not there's any of these pendulum nests that get reused through a breeding season. As far as I know, and when I've gone to like actual colonies, you find weaver nests just scattered all over the floor. They're just all over the ground. So you can tell that they're constantly making and building these things all the time during the season itself. So as far as I know, there aren't many other examples other than social weavers where they're reusing a nest after a season, right?
Yeah, that's correct, Jackie. The only other pattern that I see sometimes is you do have weavers that will build new nests on top of old nests from the previous season or near the old nests from the previous seasons. But I think it's rare that, like you said, for many of the polygynous species, it appears to be a sexually selected trait where the female doesn't mate with the male until after she inspects the nest. So it seems like, and what you said earlier in your talk, the females tend to prefer greener nesting material rather than older nesting material.
So there's a huge sexual selection component to all of this. I don't know what the parasite load is in the sociable weaver bird nest. I imagine that if they can get inundated with geckos and lizards and snakes, that the bacterial load is also pretty high because you've got a lot of animals living and breeding and defecating in the same nest. So I don't know if people have looked at parasite loads in sociable weavers. But you're right, especially in some of the more like grassy or tropical habitats, you get like nest mites, I believe, and so there's probably an added pressure of not wanting to, get a buildup of all those things, especially if you're in a gigantic colony.
You just described another postdoc project.
Got like 10 postdoc projects.
Describing nest mites of the sociable weaver.
Yeah.
All right, well, I think in the interest of time, we'll stop it there. But Jackie, thank you so much again for doing this for us. And it was a pleasure to have you, a pleasure to hear about your work. And what I would love to do, if you don't mind, is at some point, another year and a half from now or so, after you've actually gone through the rest of your variables and looked at their contribution to the phylogeny of this family, I'd love to see how much each of the other variables actually contributes to the separation among the species and subspecies. So, and see how much those new variables actually add to your ability to, discuss the relationships or infer relationships. So, but I think, thank you so much for now. And this was a marvelous conversation. It was lovely to have all of you on board here too, who have actually worked with nests and maybe even of weavers themselves, asking questions, and as well everybody else. Your questions were awesome. Thank you for your attention, and please stay safe out there, and look forward to maybe seeing you at next month's conservation series too. So thank you very much. Have a wonderful night, everybody.
And I will collect those nests in California for you.
Okay, thank you, Renee. Thank you. Once the village weavers in my backyard breed, I will call you.
Thank you, Jackie.
Okay, bye, folks.
Bye, everyone. Thank you so much.
Good night.
Thank you, Jackie.
Night.